Like Levi, oh, it's just like because you're talking about the insurance, like, and then the things he has to read, there's paragraphs and shit. No. And like, when we talk about it's then we can do this reading later. You need it to know? No, then remove it. Like you got a 12 minutes.
Okay, let's get started. All right, couple of reminders before we get started. Participation points. We have a couple of you that already completed the requirement of participation for the entire semester, so that's great. But keep answering, keep participating. I will use these as extra credit if you need them. So just continue your involvement in participation. Some of you are close. You can check with Nigel or Brittany or Jean if you want to know where you are with participation. They can show you this. But I'm worried for a couple of you that either did not participate at all or have only 2 or 3 participation. So, again, any question you might have, you can think of, any question is a valid question. Any answer, even if it's a wrong answer, I don't shame you. I don't think less of you. Nobody's going to think less of you if you don't know. You're here to learn. So let's participate. Okay, the other announcement is from Nigel. He wants you to remember that he's going to be grading lab notebooks next week in lab. So he's kind enough to remind you so that you can update your lab notebook. And if you have lost some points from the first time I graded lab notebook and you want to update that, you can just remind Nigel to look at that with you. Okay, why the middle of the class is empty? Because like I ever want to look here or look here, huh?
Outlets are over here.
Oh,
I don't know what it is. I usually, they come pre-charged.
okay. All right, so I'm gonna be looking like this or like that. Okay. All right, so I'm gonna give this back to you, Nigel. All right. Thank you. Oh, and another thing I want to let you know that this Friday, we have a guest lecturer from General Mills. Her name is Christina Wachowski. Some of you might know her already. She's an expert in IR spectroscopy. So I'm a little bit behind. I should have by today started intro to spectroscopy, but it's okay. She's a very good teacher and she will be very careful in explaining principle for you. And next week, Monday and Wednesday, because I won't be able to lecture, Gary will start the approximate analysis with moisture and with moisture determination. So this would kind of like be flopping some topics. When I come back the following Friday, I'll continue with spectroscopy. So it's just to fill in gaps where I can't lecture. We're moving things a little bit out of order. I hope it's okay with you. And if you feel lost, there are recordings that you can go back and listen to it before the midterm so that things are more in order for you. So there's nothing I can do about that. I had to move things around this semester a little bit. Okay. All right, so let's do some recap questions as we wrap up chromatography and HPLC. Let's see what you guys remember. I like to always remind you of topics so that it's fresh in your mind, and that also helps you when you study for the midterm, which will be coming up soon. How does temperature impact resolution? We talked about it a little bit. Jason in the back. So how? What is betterism? So if you increase temperature, is it like, oh, you can increase, you can run at 80 degrees, for example? You can keep increasing the temperature. Can you run at 80 degrees, for example? Yeah, so hurt the column or too fast for resolution, so you get counter effect.
I was just gonna add, we also don't want like fluctuations in the temperature, or that would impact the resolution as well.
Yes, fluctuation in temperature, that's why we have an oven usually associated with our HPLC. So we maintain a constant temperature throughout the run. Otherwise, if the temperature changes in the room, the viscosity of the mobile plates changes, either decreases or increases, impacting the retention time. Very good. Thank you, Lisa. How does flow rate impact resolution? Isaac? Yeah. Yes, very good. Very good. But also with flow rate, we have to find that sweet spot. You want to increase flow rate to enhance mass transfer, reaching equilibrium faster, preventing longitudinal diffusion, all that is great. But very fast flow rate also is detrimental in a sense. You don't give enough time for equilibrium, and also the compounds will move faster, and then they will lose the resolution and the separation. They leave the column too fast. Okay, how does the packing and thickness of the stationary phase impact resolution? Do you remember that concept? So if it is too thin, go ahead, Taylor, give it a try.
I was gonna say that, I guess the opposite, that the thicker it is, the more not longitudinal, but the other diffusion that there can be within the column. And
Yeah.
so decrease resolution.
Exactly. So you're on the right track there. So when you have a lot of stationary phase, you have chance for your compounds to go in and interact and be delayed. So they can diffuse, interact, be delayed, and then you will potentially not have very sharp peaks. And then if it is very thin stationary phase, like a thin coating or a capillary stationary phase, capillary column, you will have very fast equilibrium because there's not a lot to go in and interact with. There's not a lot of stationary media. So you reach equilibrium faster, you have really sharp peaks, you really have short retention times, and then short analytical time. But again, that's when you as an analyst, you have to think about, do I need really short analysis time, a little bit of loading on the column just for identification purposes and quantification, or do I need high capacity for my column? Do I need to inject a little bit more? Do I need to collect these fractions afterward? Then you have to choose from two different types of stationary phase, thick packing or thin stationary phase. What are the advantages of narrow diameter column, narrow and long? Go ahead, Ingrid.
We reach
Yes. So the longer your column and the narrower is the diameter, the better is the resolution, the sharper the peaks, and you get better separation. You give your compounds a chance to separate based on their affinity with the stationary phase. What are the advantages of HPLC, high performance over low pressure LC? Liquid chromatography, Jason? You can do that with both of the high pressure and low pressure. You can control flow rate, you can control the pressure, yes.
The thing is you can get reusable columns.
Very good. Reusable columns. You don't have to worry about having to pack, repack, adjust the media. The columns are already packed for you, and you can have multiple injections, at least 1,000 theoretically. But that is one thing. Can you think of another advantage? Yes, Medora?
With HPLC, you tend to like get better resolution with, your samples and it's like a little bit quicker versus like LC is better, not necessarily for resolution, but like for just separating and collecting.
Absolutely. Yes, with high performance liquid chromatography, you definitely have better resolution, better time and more controlled or purified fractions or peaks. Whereas with low pressure LC, when you're really looking at bulk and you don't really want purity and you want to separate and collect these fractions, even though they're not very pure, you go with low pressure LC. What is gradient elution in HPLC, Ian?
That's when the concentration of the mobile phase, changes over.
Absolutely. That's when you change the concentration of the mobile phase over time. What is a guard column and its function, Hallie?
The guard columns, like essentially an extra attachment that goes on top of the column and it's what removes any impurities that will be going into preventing the damage of the column.
Absolutely. Prevent impurities that might go into the column, bind strongly with your functional groups, render them not available, these function groups for the constituents you're trying to separate. So guard column is essential in HPLC. In GC, you did something different. You actually cut the column. So the beginning of the capillary column of the HPLC acts like your guard column in HPLC. You cut it off. So you take that part that was preventing or binding impurities. How many solvents and pump can one use in HPLC? Like how many pumps can you have linked to your HPLC unit? Yeah, Isaac?
3.
You can have up to 3, but usually it's either 1 or 2 pumps. 3, it becomes a little bit expensive, but you can do with 1 column and have a pre-pump mixing or low pressure mixing before going into the pump. You can have 2 pumps where you can have a high pressure mixing after each solvent is pumped through an individual pump. Sometimes you have 1 pump and 4 mobile phases. So that's very common too, or 2 pumps and 4 mobile phases. Then you have a mixture of low pressure mixing and high pressure mixing. So up to 4 I've seen that you can have in terms of solvents associated with HPLC. Very good. So let's continue where we were. We were talking about the different detectors in HPLC. We talked about the UV variable detector where you would select lambda max and measure absorption. And you can change during the run, you can measure absorption at 2 different wavelengths. Whereas the photodiode array detector, which you have used for the KFME lab, is basically you get, if it's a UV, if you're measuring under UV, the light from a deuterium lamp is spread. And we're going to learn in spectroscopy that the light diffraction, do you know what light diffraction is? You remember that from your light physics, from your physics. Give it a go, Kevin.
So when light goes through different mediums, it travels at different speeds. And so like when you put a straw in the water, you see it, the straw in the air versus in the water, light is different. They're not connected.
This is a great answer for light refraction, like refractive index. So what you were explaining is the refraction of light. Diffraction of light, I know they're confusing. What do you know the diffraction?
Would it be scattering it then?
Yes, not scattering. It's actually, I was going to use that pen. It's actually breaking the light into several wavelengths. So you have a light that has multi-wavelength light, and then it goes into either a prism or a grating system. And then when it hits, the lights kind of get diffracted into multiple wavelengths, okay? like into a range of different lambdas. And that is what happens in PDA. You get the lamp, the light source, goes into a diffraction system, either a grating or a prism, sometimes a mixture of both. And then the light gets split into multiple lambdas, and they hit an array. That's what we call it an photodiode arrays or photodiodes. And each lambda will go to one photodiode and converts into electrical signal. So that each photodiode will convert a wavelength, a specific wavelength, into an electric signal. Then you get the sample absorbs light at multiple wavelengths. Then you can measure absorption at multiple wavelengths, but of course your sample does not absorb at all lambdas. So it has a maximum absorption, and you actually get this scan every fraction of a second. Every fraction of a second, you get this scan. That's what you saw in the caffeine lab. So basically, we get kind of an what we call it a fingerprint of individual compounds that absorb light. Not all compounds absorb light, obviously, but those that absorb light, or you have derivatized in a way that the resulting compound absorbs light. Therefore, you can do more powerful identification of your compound. Okay, so we've seen that in HPLC. We also showed you that when we, actually I pulled up like the defatted coffee example, and I think Nigel did the same with the other groups as well. So you see that the beginning of the peak can be different than the end of the peak sometimes if you have coelution. If you don't have a PDA, you won't be able to see this, the impurity there within your sample. So you would see it come as one peak at a specific retention time, but you won't see that unique spectra that will tell you, this is an impurity, or you have multiple compounds coming up with the sample. So when we use a UV variable detector and we're running caffeine, we can only see, for example, like I showed you in the lab, and Nigel probably did too for the other groups, you only see a chromatographic peak. You won't be able, based on the retention time, you're gonna say this is caffeine. Whereas with PDA, you can for sure know whether or not this is caffeine or whether or not you have an impurity. So that's the power of a PDA, photodiode array detector. Okay, so refractive index detector, what Kevin was explaining, is based on the refraction of light. And we're going to talk about it in more depth when we talk about spectroscopy. So here you are measuring refraction of light as the medium changes. And this is a very common detector for carbohydrate analysis. We use it a lot in carbohydrate analysis. The only thing that we have to pay attention to is that you cannot do gradient elution with refractive index detection. Do you know why you cannot change the mobile phase during a run with refractive index? Why can't we do that? What's the principle of refractive index? Is measuring the change in refraction of light.
So I think it's the concentration of the sugars within the liquid or the mobile phase. So if you then change the concentration in mobile phase, it would change the refractive index.
Absolutely. If I'm looking for sugars or I'm looking for a specific compound, As they come out of the column, out of the column into the detector, they're gonna change the refractive index. Great, we're gonna convert that signal into concentration. But if the mobile phase I'm using is changing in concentration as well, that's gonna change my baseline reading of the refractive index. I can't do that. So that's why I would always have to have isocratic illusion with refractive index. Either that or having, I don't know if that ever happens with the HPLC. I'm just thinking if you were having some sort of reading of mobile phase alone in conjunction with reading on the sample and get subtraction that way, subtraction of the baseline of the mobile phase and when the sample comes. But that doesn't usually come with these equipment. So basically you use mostly isocratic illusion. All right, the next type of detector is electrochemical detector. So the most common one is PAD. So it has similar letters, but kind of like different than a photodiode array detector. It's PDA. So don't confuse PDA with PAD. Okay, so pulse ampermetric detector basically measure electrochemical signal. So if a compound can be oxidized or reduced, then when you run a voltage, it changes the current, and that can be detected. Or you can measure a change in conductivity of ions. So between two electrodes. So you will have electrodes, you have conductivity running, and once you have ions go through, it's going to change the reading of conductivity. So these kind of detectors, electrochemical detectors, are often used for, let's say, phenolic compounds. They can be oxidized, so they can be measured. Ions, like anion molecules or cation molecules, like amino acid analysis sometimes can be done with ion exchange using electrochemical detectors. Organic acids, carbohydrates that can be reduced, for example. Okay, light scattering detector is a universal detector. So make sure you make note of that. What does universal detector mean? I think Gary talked about universal detector, like thermal conductivity detector in GC is a universal detector. Yes, Taylor.
It has low specificity, so it can be used for a lot of different.
Absolutely, yes. So yes, light scattering detector is a universal detector. Universal detectors have low specificity, but they can detect, which makes them able to detect multiple compounds regardless of the type of compounds. So whether they absorb light or they don't absorb light, it's not a matter with light scattering detector. So it's a very powerful detector in detecting multiple types of compounds. So basically the principle is based on you have your compound traveling with the mobile phase. Once it reaches the detector, the solvent evaporates. And when the solvent evaporates, what is left is your compound of interest, and it's basically scatters the light. And based on the amount of scattering is linked to the amount of the compound. It's linked to the characteristic as well as the concentration of the compound. So it is very, very powerful for non-volatile compounds because you want to evaporate the mobile phase, but you don't want to evaporate the compounds, obviously. And it's a very powerful detector that often is used in size exclusion chromatography. So the one form of it is a multi-angle light scattering detector. So if you hear that term MALS, it's usually linked to size exclusion chromatography, helps with accurate detection of molecular weight and concentration. Okay, the last detector I have here is this doesn't make sense. This in red is for derivatization. That doesn't make sense to the LCMS. I don't know why I have it here. The LCMS, basically mass spectrometry, you already learned about mass spectrometry. It gives you powerful way of identification and detection based on molecular weight and ion characterization. Some of you already got the lab today, and you already have the lecture. Some of you will have the lab next week. So it's a powerful detector that it allows for more identification strength and molecular elucidation, especially if you have mass spec in tandem. That would be powerful detection. What I haven't read, I don't know, I think I had another point and it was deleted. It's sometimes you do derivatization. So sometimes compounds don't have any specific absorption. You derivatize it with either a fluorescent, so that it fluoresces, or a compound like ninhydrin, which is specific for amino acid composition detectant. Then you produce a compound that you can detect absorption. So the OPA is the reagent that will allow you to get a fluorescence. So you measure fluorescence instead of absorption, and ninhydrin, for example, allows measuring absorption. So this red point here, I don't know if you have it in your notes differently in your slide, but this is for derivatization, pre or post column derivatization.
And it says it's for chemical reaction.
Yeah,
So,
chemical
like-
reaction pre or post column.
Yeah, so that's what it's for.
Yeah,
But you were saying like, so is that similar to, like when they did, I mean, not similar, like analogous to
yeah,
DNA sequencing and things where like you're kind of doing something, you're adding something that you can detect onto something you can't detect.
exactly. So that's a chemical reaction. Let's say you get amino acid. Not every amino acid absorb light. Only 3 amino acids absorb light: phenylalanine, tryptophan, tyrosine. So you have other amino acids, they don't absorb light. You're doing amino acid composition and you have a UV detection. So what you would do, either a pre-column or post-column reaction with a compound. That compound can be an OPA compound that will react and give you a compound that measures absorption of fluorescence, or another like ninhydrin also gives you another compound that would fluoresce, or ninhydrin actually absorption, not fluoresce.
Okay.
Yeah. Okay, good. Let's see. So this is a good study table. It's in your chapter 13, table 1. It's kind of like when I ask you for give me an example of a mode of separation and a method of detection for a particular compound. So, for example, later on in the semester, we're going to learn about vitamins, amino acid, pesticides, all of these things. We're going to learn about the mono and disaccharides, oligosaccharides. Later on in the final, I might ask you, okay, you have pesticide. Give me an example of a liquid chromatography method that you would use, mode of separation, and potentially method of detection. So this table at the end of the semester would be a good one to go back to and review after we cover everything on that list. So a few examples. We already know what normal phase and reverse phase is. We covered that a lot. We talked about it a lot. Normal phase usually you use when you have more polar compounds that would interact with polar interactions, relatively polar interactions with your column. And we use that commonly in monosaccharide, disaccharide, oligosaccharides. We give you examples when we talk about carbohydrates, water-soluble vitamins, pesticides. In reverse phase, your interaction is basically based on non-polar, relatively non-polar interactions. So you work with compounds that might have some form of relative non-polarity. that you can use. And there's a list of examples. One here is alkaloids such as caffeine, which we used in the lab. Iron pair reversed phase. I mentioned it very briefly in one of those chromatography lectures. If you have compounds that had are charged on the surface, and you want to use reverse phase chromatography, you can add ions to pair with the charge on the molecule so that you block the charge and you can enhance interaction with the compound, with your stationary phase. These are just examples of normal phase chromatogram, reverse phase chromatograms, just FYI examples. Ion exchange, we talked about that a lot during chromatography. It can be cation, it can be anion exchange, used a lot in amino acid proteins, even mono-di-oligosaccharides. We'll talk about those examples later. Depending on the charge of the compounds, it can be cation exchange or anion. Size exclusion, it is not as common in high performance, but it is used in high performance. But it has a specific use in high performance when we're looking at molecular weight, when we're looking at degree of polymerization. We use size exclusion chromatography in high performance. So it is used for, like I said, molecular weight of proteins, carbohydrates, large carbohydrates, components. Size exclusion is used a lot in low pressure chromatography because it's kind of like one-dimensional separation of compounds based on size. Affinity chromatography is very specific. It has very specific use. So if we are purifying enzymes, antigens, antibodies, mycotoxins, you can then use very specific media that has very specific interaction with your compound of interest. Mostly used for purification. These are just FYI, again, example chromatograms. In size exclusion, you can see that often your peaks are not really sharp. They're kind of like overtones, just because a lot of molecules have very similar molecular weight. So you can estimate size, molecular weight based if you have standards, but mostly if you want to do it as a first dimension separation before you do further purification. All right, let's do an exercise. I want you to pull out a piece of paper, write your name on it. This is for a +2 in class exercise. And I like this question very much. You might see it in a quiz, you might see it in an exam, it might come anywhere. So I have several questions. Just put your name on that paper. Put example chromatogram questions and put number 1 through, I don't know how many questions I have, 1 through 5 answers to the questions. Okay, so this is a chromatogram that I showed some of you in the lab and Nigel showed the rest of you of isoflavones. Okay, you have different isoflavones that are present in soy, and this is a mixture of standards. And this actually I worked on when I was a postdoc. So I developed a method to separate these isoflavones. And this method was based, well, I'm not going to say it because it's one of the questions. So I separated those different isoflavones. This is the gradient used. I start with 17% acetonitrile at 0 time. And then it goes up to 25% acetonitrile. Then it stays idle or isocratic for 5 minutes, goes up again, gradient to 30%, stays isocratic for about 10 minutes, and then in 2 minutes goes back to 17% and stays at 17% for another 5 minutes or so. It's basically equilibrating the whole HPLC to go back to the original concentration of the start so that I can inject another sample. All right, so now that you know what you're looking for, is this normal phase or reverse phase chromatography based on this, the start, 17% acetonitrile, 83% water. So is this normal phase or reverse phase chromatography? Go ahead, Trina. Huh? It's reverse phase. Thank you. It's reverse phase because it's 83% water, so you have more polar. And then it increases in acetonitrile concentration. That means polarity decreases over time. So this means we are starting with reverse phase chromatography. So you have a relatively nonpolar column and a relatively polar mobile phase at the start of my run. Okay, very good. Which of these isoflavones is the most polar? The first one or the last one? Most polar. Go with me.
The first one.
Yes. Yes, in reverse phase, the most polar compound will have the least interaction with your column. So it will elute first. And the last one would be the most polar because it will interact the most with your column and will only come out when you really increase this acetonitrile concentration. Okay, how would you change the mobile phase? So genistein, which is the one that elutes last, the last one would elute sooner without changing the relative position of the other compound. So what is this asking you? Sorry. What is this asking you is how can I make this compound that is about 30, from 35 maybe, or even less, till about 49 minutes or so, there's nothing. And I'm waiting about over 10 minutes here for this to come out. So how can I change, what do I change in my gradient to bring this sooner, but not affect any of those? All right, go ahead, Kevin.
Some point after the last one there, that's not the actual last one, but in between,
Yeah,
then you can start increasing the acetonitrile.
even more than 30%.
Yeah, maybe like 35.
Exactly. So after this 32, 35, let's say to be on the safe side, after 35 minutes, I don't, or here when I reach to 30, I don't necessarily have to keep this idle. I can, or isocratic, I can continue to, let's say, 35, 40%, and then this will come sooner. And I won't impact any of my other compounds. I don't want to change. I have perfect resolution. I don't want to cause any co-elution. Very good. All right. So what would happen if you keep an isocratic elution mode at 17%? What would happen if you decide to run at 17%? Only, Medora.
Your number one, the peaks, the resolution we have here as well is good, and number two, it will just take a long time.
Absolutely. With resolution not being as good, that means you might have, especially those peaks that are close to each other, you would have a lot of diffusion within a lot of interaction within the column, your peaks might become broader. So if the peaks get broader, we might see this as a singular peak, these three as maybe two peaks and not very well separating, same with those three. And it will take maybe 4 hours for all of them to come up, especially this last one. And I might just get tired, stop the column and stop the run, assuming that everything came out, but really some residues might have stayed on the column, which will impact my next run in terms of contamination. So that's a very bad idea, really, when you have compounds that are very, very nonpolar. Okay, what would happen if you keep an isocratic elution mode at 30%? Go ahead.
Would there be more like pollution with them because then there's less time for it to create like those strong heats?
Absolutely. I'm reducing selectivity. You remember, always remember your efficiency, selectivity, and capacity. I'm really reducing selectivity because now at 30%, everybody would want to come out with the mobile phase. So you will lose selectivity, you will have co-elution, and everything will come out sooner, and it's not effective. Okay, so these are the things we think about when we're developing separation. Is do I want gradient? Do I want isocratic? If I do gradient, how do I have a balance between good separation and shortest analysis time? So things that we think about when we develop our separation protocol. All right, last thing I want to mention here in liquid chromatography, in HPLC, we can do sequential chromatographic separation. 2-dimensional, 3-dimensional, sometimes 4. This is an example where we can use multiple HPLC separation to purify a certain, in this example, peptide. So in this example, I have generated, I took my proteins, I hydrolyzed them with enzymes, generated a mixture of small molecular weight peptides, large polypeptides, and I have this mixture, and I know in this mixture now I have peptides that are bioactive, have antioxidant activity. So I want to purify for pharmaceutical reasons. I want to purify the most potent peptide, and I want to sequence that most potent peptide. So the first thing I would do, ultrafiltration. You're going to learn more about it when we talk about protein characterization. But in ultrafiltration, you basically have a membrane with different pore size, small pore size. So you separate the large polypeptides from the small peptides. So the first thing you do, you just take out all the large peptides because you know that your bioactive peptides are going to be small. So we get rid of large molecules or polypeptides, and then we can run first dimension chromatography. It can be size exclusion followed by ion exchange, or the other way around. It can be ion exchange followed by size exclusion. It doesn't matter really the order. You can pick one. Usually, actually, it's the opposite of what I have here. Usually, you start with size exclusion, then ion exchange. So what you do is you separate based on one or the other. You get fractions. You collect the fractions and measure your bioactivity, say antioxidant. You select the fraction that has the highest bioactivity, and you take that fraction and you inject it on the second dimension HPLC based on size. And you also take the fractions and measure the bioactivity. Then you go from there and you go to a third dimension separation, let's say reverse phase chromatography. And then you get, again, different fractions. You take that one that has the highest potency, You run it with MS, you get very pure peak with the most potent bioactivity. Then you can send it and do more sequencing analysis on it to determine amino acid composition. So HPLC can be very powerful tool for so many different applications. And that's one application where you can use HPLC in tandem. All right, I have 5 minutes. Great, I will start spectroscopy, just the intro part. Okay. All right. Lots of information, right? Okay. This is the last part of this class that is based on principle of, methods, chromatography, spectroscopy. And then we're going to start actual, composition and analysis, characterization of different components and so on. So this section requires a little bit of remembering your physics, if you liked physics, and especially the physics of light. And so I'll go over principles that are essential for you to understand spectroscopy, but hopefully you will remember your previous physics knowledge, so that will be easy. All right, so let's think about a light wave. So what do you remember? The following are associated with a light wave. You want to give it a go?
No.
I thought there was like some hesitancy. Trina, go for it. A and C. Okay, good, but any other opposition or other ideas? What did you want to say, Isaac?
It's at least C.
It's at least C. Okay, so he's on the safe side. At least C is correct. Just C? Okay.
I think it's all.
Kevin, yes. Have you watched Analyze This? Do you know this movie? It's a very old movie. You're good, right? Remember that part? What's that?
I like light. When I was younger, I used to.
Yeah.
Yeah.
So you're all, I'm gonna say you're all good, you. Okay. I'm not favoring Kevin here. All right, so it is all of the above. You have electric field and magnetic field. We'll talk about those. And photons, it's kind of like if you want to think about the light as a wave or as a particle. So the photon is the particulate of light that carries the energy. So you can differentiate light wave with electric and magnetic field, but at the same time, you can define the light wave by the pockets of energy. So by the photons, which are defined as pockets of energy. Okay, so all of the above. All right, so following spectroscopy as means of analysis, detection can be based on absorption of a photon, which is particulate of energy, emission of a photon, dissipation of heat. When you use spectroscopy to detect, quantitate, do you usually base it off of absorption, emission, or of light, or a photon, or dissipation of heat? No, don't be shy. You don't know this or you don't remember this, but give it a go. Go ahead. A and C. Okay. Any other answers? Go ahead. A and B. All right. Any other answers?
Sorry, I was about to say A and B. I meant to be on the.
A and B? It's A and B. All right. So, the excitation to the excited state and then going back to ground state, that's when you absorb energy, when you take on energy, you go from a ground state to excited state. And then when you go from excited to ground state, the compound may emit a photon of light that can be measured. Not every compound can emit a photon of light when going from excited state to ground state. But in almost all cases, there is a small amount of heat that can be dissipated when you go from ground state to excited state. Sometimes it's only heat. Sometimes part of it is photon, is emission of a photon and some heat. No matter what, if it's all heat or a little bit of dissipation of heat, it's a very, very small amount that cannot be measured. So cannot be measured in sensitivity. So we do not use C. It happens, it occurs when you go from excited state to ground state, but it's not in a measurable amount. Okay, so there is absorption of light, there is emission of light. Okay, so spectroscopy basically involves three things. Species, I want to just cover this and let you go. Give me 2 minutes. This will prepare you to Friday. So species, so what am I measuring? Am I measuring a molecule or am I measuring absorption or emission of light by an atom? Like some of you did atomic absorption. So you're either measuring absorption or fluorescence of a molecule or an atom. And then you have, in order for spectroscopy to occur, you need radiation matter interaction. You need them to interact. So the interaction is either the molecule or the atom is going to absorb the energy of light, or it has the ability to fluoresce, that means emit light, or it doesn't absorb, it doesn't emit, it's going to scatter the light, or it's going to diffract the light. So all of these principles of absorption, emission, light scattering, diffraction are important as we talk about the different types of spectroscopy. UV visible, IR, some instruments in IR rely heavily on diffraction. So that's what you will learn with Christine on Friday. And then you have different wavelengths that are relevant to us. And then when you have the wavelengths, as they become smaller, they carry more energy. And I'll talk more about that next week. So the smaller the wavelengths, the more energy it carries. And what we care for in fluid analysis, the applications are in UV, in visible, and in infrared. You will cover with Christine with infrared and near infrared, MIR and NIR. So, and then this will be covered with Christine and she'll give you the principle, but then I'll come back, talk about more about UV, visible, absorption, and emission. So cover both when I come back. Okay, so good luck. You will be armed with a little bit of knowledge before Friday.
Yeah, I'm turning these.
Yes, give me the extra credit or give them to Nigel, right?